Dear learners, a warm greetings. This is Dr. N. Jeenathunisa, Assistant Professor in PG and Research Department of Microbiology, Kaveri College for Women, Autonomous, Tiruchirappalli. In this e-content, let us have an overview about the bacterial cell structure and functions. Bacterial cell have a simple structure. It possess both external and internal structures. The external structure is present outside the cell wall. It is the capsule, flagellum, and pilus. The internal structure have cell wall, plasma membrane, cytoplasm, nucleoid, ribosome, and plasmid. Let's see about the capsule. Capsule is otherwise called as the glycocalyx layer, is a gelatinous covering with high molecular weight. This viscous layer has thickness of about 0.2 micrometer. There are two types of capsule, namely macrocapsule with a thickness more than or equal to 0.2 micrometer, and they can be seen only under light microscope. The second type is the microcapsule. It has a thickness less than or equal to 0.2 micrometer, and this can be visible only under electron microscope. The function of the capsule includes attachments, prevention of cell from desiccation and drying, and it also resists phagocytosis by WBC cells. Next comes the flagella. Bacterial flagella is a hair-like structure that emerges from the cell wall and measures about 15 to 20 nanometer. The flagella is composed of the protein called flagellin protein. It is also known as H antigen. Bacterial flagella have three parts, namely the basal body, hook, and filament. The flagella helps the bacteria in motility. Here we can see about pili or fimbriae. Pili is a hollow filamentous structure which is non-helical and found to be numerous and shorter than flagella. Pili is a characteristic feature of the gram-negative bacteria. It is composed of pilin. This pili helps the bacterial cell in attachment, and it also has some antigenic properties. They have special functions, as some of the pili are modified as F pili or sex pilus, which helps in conjugation. Next, we can see about cell wall. The cell wall is present internal to the bacterial structure. It is a most important part of bacteria as it gives a definite shape to the cell. Based on the cell wall composition, bacterial cell wall can be gram-negative, which has a plasma membrane and a thin peptidoglycan layer. Between the plasma membrane and the thin peptidoglycan layer, there is a periplasmic space. It also have an outer membrane made up of lipid, protein, and lipopolysaccharide layers. Next comes the gram-positive cell wall. It has plasma membrane and a thick peptidoglycan layer. A periplasmic space is present between them. Now we can see about the cell membrane. The cell membrane is also called as cytoplasm or plasma membrane. It lies under the cell wall and encloses the cytoplasm. The thickness is about 80 nanometer. The cell membrane is composed of phospholipid and protein. The main function is the selective permeability. Now we can see about cytoplasm. Cytoplasm is a colorless, viscous fluid present inside the cell membrane. All the cell organelles and inclusions are found floating in the cytoplasmic fluid. It contains protein, lipid, minerals, nucleic acid, glycogen, water, and so on. Now we can see the nucleus of the bacterial cell. The bacterial nucleus is also called as nucleoid. It is the most important part of the cell, and this nucleoid controls and directs all the cellular activities. It also stores the hereditary information of cell, and it also happen to be the site for transcription and replication of DNA. Next comes the ribosome. The bacterial ribosome is of 70S type with a larger subunit of 50S and a smaller subunit 30S. Ribosome is known as a universal cell organelle because it is found both in prokaryotic and eukaryotic cell. The main function of ribosome is protein synthesis. And the last internal structure of the bacteria is the plasmid. Plasmid, it was first coined by Joshua Lederberg in 1952. Plasmid is an extrachromosomal DNA which can replicate independently of the host cell. The main function of the plasmid is to carry the antibiotic resistant gene. As learners, from this e-content, we can have a clear overview about the bacterial cell structure and functions. Hope to connect with you all in my next e-content. Thank you. Have a nice day.